This is Dangers of Nanoparticles by Kimberly Villagran, Mia Seha, Mia Martinson, and Melissa Herrera. I'm Kimberly and I will go over the background. So, what are nanoparticles? They are extremely small particles ranging from 1 to 100 nanometers in size. To put into perspective, they are smaller than cells, bacteria, and some viruses. Types of nanoparticles are metals such as silver, gold, titanium dioxide. Carbon-based nanoparticles include fullerenes and carbon nanotubes. Polymeric nanoparticles such as synthetic or natural polymers formed at the nanoscale. Ceramic nanoparticles include silica and alumina particles. They are made by natural processes such as volcanic eruption, forest fires, and sea spray. They are also made by anthropogenic processes such as industrial production, construction, mining, consumer products, waste, and pollution. Finally, engineered nanoparticles, such as purposely designed for specific applications and modified properties, are all types of ways nanoparticles are made. Some characteristics are that they have increased surface area, ability to pass through cellular barriers, interactions with subcellular structures, increased ability to activate neutrophils, and the simulation of the release of inflammatory mediators.
So, why study nanoparticles and what are potential concerns? One of the biggest reasons why nanoparticles are increasing in popularity is due to their effectiveness against bacteria and viruses, potential use as biocides, and use in technology, along with many more functions. One of the reasons why they are useful against bacteria and viruses is due to their high surface to volume ratio and their unique chemical and physical properties. They are also useful for technology due to their high surface to area mass ratio, large quantum effects, deformability, durability, tendency to aggregate, optical sensitivity, hydrophobicity, high reactivity, rapid dissolution, electrical conductivity, and great tensile strength, which is also one of the reasons why they are so toxic to the biological systems. And one of the potential concerns is that as the rate of nanomanufacturing research is accelerating, there are more increased amounts of nanomaterials in waste. This is very dangerous because there is abundant evidence to show that the association of levels of inhaled particles with the elevation of cardiovascular mortality and morbidity. One research by Song in 2009 found that in China, when 7 workers in a print factory were exposed to a polyacritic ester paste that contained nanoparticles, the workers developed an unusual and progressive lung disease in which 2 of them died from that. And much of the current nanoparticle exposure is due to consumer products such as makeups and sunblock. And a lot of the nano waste stream examples, ranging from different classes of toxicity, include the display back panels of TV screens, polishing agents, paints, food packaging, food additives, personal care products, sunscreen lotions, pesticides, and many more. So, why would we continue to study nanoparticles? One of the biggest things they are hoping to achieve is the possibility of helping detoxify drinking water supplies, which is very great advancement since the lack of drinkable water is one of the greatest issues worldwide. So, the specific research questions being asked with these advancements are: what are the long term effects of nanoproduct exposure? How can research continue to be done without increasing the buildup of nano waste particles? And how can we remove the nano waste from the current products used on a day to day basis?
In the study, E. coli was chosen because it has no plasmids, which ensures that any resistance is due to mutations. Scarcity of known silver helps in this experiment because it helps to identify new genetic adaptations that confer resistance to silver nanoparticles. E. coli was cultured in minimal media with dextrose and thymine, with transfers to establish growth. Control groups grew without silver nanoparticles, while treatment groups were exposed to increasing concentrations of nanoparticles over multiple generations. Bacterial growth was measured by turbidity and colony counts and minimal inhibitory Concentrations were determined by performing serial dilutions and finding the lowest silver nanoparticle concentration that inhibited growth. DNA was extracted from selected generations and sequenced to identify genomic mutations associated with silver nanoparticle resistance. This allowed the study of evolutionary resistance developed in E. coli. The main result of the experiment was that E. coli developed resistance to silver nanoparticles after exposure to increasing concentrations over many generations. This resistance evolved through genetic mutations. Table 1 shows the key results as it lists antimicrobial resistant genes in E. coli and shows that the strain lacked significant resistance loci. It explains that the observed resistance to silver nanoparticles was likely due to mutations and not because of pre-existing resistance mechanisms. It also showed that E. coli evolved very rapidly.
This study aimed to investigate how quickly E. coli could develop resistance to silver nanoparticles and to identify the genetic mutations associated with this adaptation. Contrary to earlier assumptions that bacteria would struggle to resist metallic nanoparticles, E. coli rapidly evolved resistance. By generation 162, treated populations exhibited increased resistance to silver nanoparticles, reaching significant resistance levels by generation 250. Genomic analysis revealed that by generation 200, multiple mutations associated with the silver nanoparticle resistance had become prevalent. Notably, mutations in the CUSS, PURL, and RPOB genes, which play roles in heavy metal regulation, purine biosynthesis, and gene expression, respectively, were linked to this resistance. These results suggest that bacteria can adapt to silver nanoparticles through relatively simple genetic changes, highlighting the need for caution and using nanoparticles as biocides due to potential environmental impacts on microbial communities. After reading the two papers, some key takeaways that stood out most to us were the following. Benefits versus costs of nanomaterials. One of the most interesting takeaways was learning about the contrasting nature of nanomaterials. These materials have unique chemical and physical properties such as high surface area to mass ratio, deformability, and reactivity, which make them valuable in fields like electronics, medicine, and environmental science. For instance, their tiny size allows for high precision in medical treatments, while their reactivity enables them to interact with other molecules in unique ways. However, these same properties pose significant risks to biological systems. The high reactivity and small size allow nanomaterials to interact more easily with biological cells, potentially penetrating cell walls or binding to harmful environment pollutants like heavy metals. This duality makes it hard to harness nanomaterials' benefits without increasing its toxicity risks. Understanding this balance is essential to prevent potential health hazards as we continue integrating nanomaterials into everyday applications. Graves also emphasizes the evolutionary perspective. When researchers study toxicology or how chemicals affect living things, evolution is very important to consider. Evolution helps us understand how different organisms might respond to chemicals over time. We apply evolutionary thinking to nanomaterials, meaning we're asking how living things, whether that's animals, plants, or microbes, might react to these new particles, and if there's a chance that they could develop resistance, or if exposure could cause unexpected effects on a biological level. This perspective, as highlighted by Graves, pushes us to think carefully about the broader impacts of nanomaterials on health and safety. Lastly, a critical takeaway is the urgent need to prioritize nanosafety research. Nanotechnology is advancing at a rapid pace across industries, from information technology to food science. However, the rapid adoption of nanomaterials hasn't been matched by adequate research into their safety. This imbalance raises serious questions about the adequacy of current nanosafety guidelines, as there may be risks that we're not yet aware of due to a lack of testing and long-term studies. Given the extensive use of nanomaterials and the integration into products that impact our daily lives, the need for a proactive nanosafety research agenda is more pressing now than ever, as stated by Dr. Graves. Without it, there's a risk of overlooking significant health and environmental consequences, which can undermine the benefits that nanomaterials bring.
So now we're going to talk about nanoparticles and its relationship with evolution. Some key concepts of evolution that were mentioned in these articles were selective pressure on E. coli was induced as an evolutionary response. Silver ions are normally susceptible to bacteria, but adaptations occur quickly and then they become resistant to it. Also, mutations introduce genetic variation. This research article illustrates how mutations lead to resistance in E. coli. Finally, organisms, fitness, and trade-offs of E. coli with silver nanoparticles resistance have improved fitness in a silver nanoparticle environment, but have a reduced fitness when growing in normal conditions, where nanoparticles are absent due to protective proteins that help remove nanoparticle detract from other cellular processes, and this is a trade-off. But understandings that we gain from these articles were that organisms can quickly adapt when under strong selective pressure. Mutation is random but crucial component of evolution, and adaptations are balanced between competing factors, making evolution a nuanced process.